They're very, very important in recycling waste products and dead organisms through decomposition. They actually nutrients from food during digestion from our gut microbiome, and they actually protect us from becoming infected with harmful microbes. So microbes are, generally speaking, our friends, but not always. So now I'm going to tell you a little bit about what we're doing about some microbial diseases. So what are we doing about Kinetoplastid diseases? And a very reasonable question would be, what the hell are Kinetoplastid diseases, and why are they important? Well, really, there are four main Kinetoplastid diseases that we need to worry about. Two of them are very, very closely related and caused by parasites which are virtually indistinguishable, and both are transmitted by the tsetse fly. One causes human African sleeping sickness, and the other causes a disease in cattle called Nagana. Now, this is a really horrible disease. This parasite gets into your blood when the tsetse bites you, replicates inside your bloodstream, and then eventually some of the parasites get into your brain, and that's when the problems really start, because the symptoms of this disease are character disintegration, coma, and death. And it's invariably fatal if it's not treated, and the existing drugs are extremely poor, very, very toxic and very difficult to administer. I'll come back to that point a little bit later. The leishmaniases are a complex set of diseases, but visceral leishmaniasis, where the parasite tends to occupy and replicate in your liver and spleen, is caused by this little tiny biting bug, the sandfly, which transmits the leishmania parasite, which chooses to invade and live inside your macrophages. Now, your macrophages are those giant white cells that normally go around and munch up on microbes and kill them. This organism has learnt a way of stealthily invading the macrophage without activating it, and then lives and replicates inside the very cell that normally clears infection. And this causes a very, very serious systemic disease called visceral leishmaniasis. And then in South America, South and Central America, we have Chagas disease, caused by these biting reduviid bugs, and here you see the parasite, very similar to the African trypanosome, but the South American trypanosome doesn't stay in the bloodstream. It actually burrows and invades our own tissues, particularly heart and smooth muscle tissues. So these are all very, very serious diseases. Just one anecdote about Chagas disease. It's named after Carlos Chagas, who is a very famous Brazilian scientist, and it was actually commemorated by putting him on the 10,000 Cruzados banknote, but sadly, because of hyperinflation, this banknote only lasted about 3 or 4 months, which I think is a great tragedy. Anyway, it's just to point out that Chagas disease is CH, because some people confuse it with sexually transmitted diseases, but it's named after Carlos Chagas with a CH, very important. Okay, so why do we need new drugs for these Kinetoplastid diseases? Well, the existing drugs are either very, very toxic, or they're being lost to us because of drug resistance. And this theme of drug resistance will come up several times tonight. This is something which we, as a species, really need to concentrate on, which is to replace drugs as we lose them to resistance. So, for example, sleeping sickness is treated with these drugs, milsaprol and pentamidine. Milsaprol, ARS, A-R-S. Why? It contains arsenic. This is an arsenical compound. This is still a frontline drug in sleeping sickness treatment. It kills 1 in 20 patients on first administration. Stone dead. Can you imagine that being acceptable in the developed world? A drug that kills 1 in 20 patients. It's very, very toxic, and even we're also losing it to drug resistance. There is a drug combination, nifurtimoxiflornithine, which has replaced most of these other drugs, except in certain cases. But look at this, you have to take 2 intravenous infusions per day for 7 days. So although this is a much safer drug combination, it's not without toxicity incidentally, administering that in the Democratic Republic of Congo is not the easiest thing in the world. And indeed, the kit to treat 1 patient is a huge package, about this high and about ½ a meter in each dimension, that has to be shipped in order with all of the IV systems to treat a patient. So not very practical. In leishmaniasis, pentavalent antimonials, antimony-containing compounds. I mean, this is just stuff from the Stone Age. very, very toxic. And then in Chagas disease, benznidazole, very toxic again. 30% of patients can't complete the course because of kidney toxicity. So bad set of drugs. We need some new ones. So my colleagues and I set about trying to do something about this directly. And about 11 years ago, we set up a thing we call the Drug Discovery Unit, which has grown to today is a team of 95 staff, 95 scientists, all dedicated to doing what we call translation, taking scientific knowledge and discoveries and trying to convert them into new medicines. So I'll give you a sort of timeline for what we've been trying to do in kinetoplastid diseases. So all of this started in 2002 when myself and my friend and colleague Alan Fairlamb were walking through the lab, and Alan looked extremely glum. I said, "What's the matter?" He said, "I've just been collaborating with a pharmaceutical company who will remain nameless, and they've just done a screen on an enzyme I discovered to try and make a new drug. But there's been a change of policy in the company, and they won't even tell me what the output of the screen was. The whole project was dead." So we talked about this for a while, and we decided, well, if we can't take our problems to the pharmaceutical industry, and they're not interested enough in developing medicines for the very poorest in the world, then maybe we should in-source those scientific methodologies that industry has into the university. And that was quite a bold thought back in 2002. So we set about doing that, and by 2005 we'd built some new laboratories to try and do that, and more importantly, we'd recruited some very, very key individuals. Paul Wyatt, a very experienced drug discoverer from industry, joined us from, he worked at GSK and then Astex Therapeutics, and joined us to direct our drug discovery effort. Ian Gilbert, our head of medicinal chemistry, and Julie Frierson, our head of drug screening biology. They joined us in 2005, and we got going in a big way in 2006 with an initial grant from the good old Wellcome Trust, which is a biomedical charity based in the UK. It's actually the second largest medical charity in the world, spends about £1 billion a year on biomedical research, and they were very, very kind to give us a large bolus to get started. Well, by 4 years later, we'd actually made a drug candidate for human African sleeping sickness. It failed, but we made one anyway. So just to point out that this is a tough game making new drugs. There's a lot of what's called attrition, where you take 5 steps forwards but 6 steps backwards. This was one such case where we delivered what was really a very good drug candidate against African trypanosomiasis, but it lacked one key property, which was it didn't get into the human brain to treat the second stage of the disease, so we had to scrap it. Nevertheless, it was a great proof of concept project because it showed that we could actually produce good pharmaceutical drug-like compounds in our academic setting, because there was a lot of skepticism at the time about whether that would be possible. So in 2011, we were awarded another dollop of cash from the good old Wellcome Trust, and this team of people, Paul Wyatt, Ian Gilbert, David Gray, Kevin Reed, myself, and Alan Fairlamb, embarked on another sort of 5-year program of discovery. We brought in 2 other key molecular parasitologists, David Horn and Mark Field, in 2013. And the most important output was that by 2016, by the end of last year, we delivered 2 what are called preclinical drug candidates for visceral leishmaniasis. Now, this was done in collaboration with GlaxoSmithKline. So it's interesting how things change. Back in the early 2000s, so 2000, 2001, 2002, it was quite difficult to engage with major pharma on the quest for drugs for neglected diseases, for a very simple reason. The pharmaceutical companies are beholden to their shareholders, and shareholders include people like me, right? I have a pension plan. I'm a shareholder in Glaxo and Pfizer and all of these companies, and therefore they need to make a return. So saying we're going to spend billions of pounds making medicines for people who have no money to purchase them isn't a very good business model. So we have to collectively, as a society, come up with other business models to make this happen. Things have moved on a lot, and now companies, particularly GSK, are doing a fantastic amount of work to helping develop drugs for neglected tropical diseases. but it's because there are downstream partners like DNDi, Drugs for the Neglected Diseases Initiative, based in Geneva, who receive core funding from the Gates Foundation, from DFID, that's the Department for International Development of the UK government. I have to say, UK citizens should be very proud of what the UK does in this space. We're one of the major donors to international efforts like this. The Wellcome Trust and others are also investors in DNDi. So now we have a possibility of working with our pharmaceutical company partners to produce candidate drugs, which we then hand over all of the intellectual property, the patents and everything, to a downstream partner, Product Development Partnerships, they're called PDPs, based in Geneva, totally charitable mission, and their job is to see whether or not those assets are good enough to go through clinical trials and then put them through clinical trials and hopefully get them to patients. So this is only halfway along the journey. These are clinical candidates. They now need to go through safety testing and probably will enter clinical trials probably towards the end of next year. But both of them are looking really exciting as possible therapeutics for visceral leishmaniasis. Now, when you want to eliminate a disease, you need a number of different things. You need to have accurate disease surveillance. You need to know where the disease is. You need to have, therefore, appropriate diagnostics. Otherwise, you don't know where the disease is. You need safe and efficacious vaccines and or safe and efficacious drugs. And you need a robust healthcare system to deliver them. And a lot of countries don't have a robust healthcare system. So there's a lot of work to be done in the delivery area. They always say there are three problems in drugs: discovery, development, and the last D, delivery. So getting existing drugs to people who need them is still a major problem in many parts of the world, particularly sub-Saharan Africa. But there are many good organizations working on trying to solve these problems in parallel. Because what you don't want to do is to solve the discovery problem and then solve the development problem, only to find you've got a delivery problem. So I'm very pleased to say that a large number of organizations worldwide are working to solve all three of these problems in parallel, so that now, as new drugs are becoming available, they are being delivered to the patients who need them in record time. So things are actually not as bad as they used to be, say, a decade ago. Anyway, why am I telling you all this? I want to just mention very briefly something to do with diagnostics, which we have made some small contribution to. So this is different from drug discovery, but it's diagnostic development. So using some quite high-tech capabilities and technology, a thing called quantitative proteomics that uses high-end mass spectrometers, a PhD student of mine, Lauren Sullivan, created this little device. It's a bit smaller than this. It's only about 3 inches long, but it looks rather large on the screen. So it's very much like a pregnancy test device, about the same dimensions. But here you put a spot of blood on these test pads, followed by a couple of drops of buffer. And if you're not infected, you just get the control test complete band lights up at the top. But if you're unfortunate enough to be infected with human African trypanosomes, you will give two test bands will come up in pink as well. So very high-tech technology to develop the device, very low-tech device for use in the field. So these cost about 50 US cents each, okay, to diagnose in the field African sleeping sickness. And you don't need any equipment. You don't need electricity. You just need a pair of eyes, okay? Very, very simple device. And here you see Lauren giving David Willett a prize, which I thought was really nice of her. So it's a good example, I think, of taking some basic molecular parasitology, some advanced technology, industrial collaboration with BBI Therapeutics, who are our partners on this. Very important, actually, was the statistical analysis. Mathematicians played a very big part in this project. But most importantly, the dedication and talent of Lauren and her colleagues, delivering impact for patients. So this is being taken forward into the field through another organization based in Geneva, another not-for-profit organization called the Foundation for Innovative New Diagnostics. And this is from their website, saying that they're working with the University of Dundee, finally is working to drive down the price of testing. So we're very pleased that this has been through field trials and should be the frontline diagnostic from 2018 onwards. Okay, so that's Kinetoplastid diseases. What about malaria? Now, malaria is a much more famous disease, and for very, very good reason. So each year, about 200 million people become infected with malaria from mosquito bites, and it still kills around about 500,000 people each year, mostly children under the age of 5 years of age in sub-Saharan Africa, but also it wreaks tremendous hardship in Southeast Asia. Now, the main frontline drug, which was discovered and developed in China, is artemisinin, and it's a fantastically good drug. It's saved millions of lives. But resistance to artemisinin is currently sweeping across the Thai-Myanmar border as we speak, and there's a huge need for new anti-malarials to be created to replace artemisinin as we use it. Now, I'm pleased to give you a little respite and show you a few interesting slides. These are from Pearl Gan, who's in the audience, and she's been working with Kevin Baird at the Oxford University Clinical Research Unit in Ho Chi Minh City in Vietnam, and with the support of the Wellcome Trust. And she's been taking some wonderful photographs, which really, I think, show the impact and effects of malaria in parts of Southeast Asia. And these are just a few of her images. And as she quite rightly says, she's going to have an exhibition of these shortly in Singapore. And the part of the reason is in order to elevate awareness of this problem among the urban elite of the Asia Pacific. I think that refers to all of you. So be aware and become aware by going along to the exhibition, which is going to be between the 2nd and 29th of September at the National Library. So please go along to see Pearl's beautiful work. And I now get 10% of all the ticket sales. So, so that's all right then. Okay, so back to malaria. So this is the incidence of drug-resistant, artemisinin-resistant malaria in Southeast Asia. And the redder it is, the worse the situation. And so you see there's a wave of drug resistance in the malaria cases, which is sweeping in this direction. This is exactly what happened between the 1960s and 1990s for a drug called chloroquine. We used to have a fantastic drug for malaria called chloroquine, and now it's completely useless. Resistance started in Southeast Asia, particularly in Thailand, and then moved through the Indian subcontinent and into the African continent. And by 1990, this wonderful drug was completely useless. And the same thing, I'm afraid to say, is starting to happen with artemisinins. It'll take several years before they're rendered useless, but unfortunately, they will go. So we need to have some replacements ready. So that brings in an organization called the Medicines for Malaria Venture, another one of these PDPs, Product Development Partnerships, based in Geneva, a not-for-profit organization who broker, if you like, collaboration between academics and industry to come up with a whole portfolio of new drugs and get them to people who need them. It's a wonderful organization. I have to declare an interest because I sit on the board of MMV, but it is really a remarkable organization. So in this story, the same team here, back in 2009, worked with MMV, Medicines for Malaria Venture, to do a little screening exercise. We grew the parasites, and we took a library of only about 5,000 compounds in this particular case, very small library, and asked which of these 5,000 compounds kill the parasite. And a few did, and so some of those were developed further into what we call drug leads, and then further again into a drug candidate. And that was we delivered that to MMV again, gave all of the IP, all of the patents to MMV, and they then partnered it with Merck KGaA, which is one of the world's largest pharmaceutical companies, because through their public good program, they are going to pay for the development of that molecule through safety trials, and it's just about to go into human beings, into clinical trials in July of this year. So that's quite a good timeline for our development of antimalarials. It got a fairly good press when it came out, but the best thing to do is to I just think, do I get sound on this? Can I get sound on my computer? I should have asked for this earlier, but didn't. So if the text at the back is, can I get sound on the computer? If not, I'll skip the little movie, but we'll see how we go. Yes? Okay, I'll plug it in then. Ooh, whoa. Let's see whether the movie works or not. Oops. So let's see if we can hear from the people who developed the compound themselves. That doesn't sound too good. No? Yeah, it is fully inserted. I think we're going to have to skip that. That's my fault for not organizing this earlier. No? Okay. We'll skip that. Okay, which is a shame because it was a little 3-minute video of just comments from my colleagues who developed that medicine and their sort of sense of motivation. But suffice to say, it requires the input of a very large number of people to develop a new candidate drug, and working together from lots of different scientific disciplines. So what we did in this particular case was we did what's called a cell-based screen. We took the parasites themselves, screened them against lots of compounds, got cell hits, compounds which killed the cells in culture, and developed those through to leads and through to preclinical candidates. And that sounds all very, very easy, but there's a lot of problems involved with doing drug discovery because it is a multi-parametric optimization problem. That sounds really bad, and it is really, really bad, because you have to do 4 things simultaneously. So at every stage you want to increase the drug potency, i.e. increase the desired effect of the drug. You want to increase the drug solubility, because if a compound, new drug isn't soluble in water, because we're made of water, it doesn't get into the body. And that's actually one of the most difficult things, is to get a very potent compound which is very water soluble. And we need to reduce drug toxicity, the undesired effects, the side effects of the medicines. And we need to adjust drug metabolism, how quickly the body eliminates the drug. Generally speaking, we want to make that as long as possible. So we've got these 4 parameters that we need to optimize at the same time. And many of them are mutually incompatible. So the old folks in the audience will be familiar with the old ball bearing game. This is exactly the problem, where you've got multiple ball bearings and you have to get them all into the center at the same time. And you've got 3 in place, and then you try to get the 4th one in and the other 3 pop out. It's exactly that problem, multi-parametric optimization. This is the iPhone equivalent for the younger people in the audience. So that's why that one's round, that one's square. But that's the problem that we have. But this business of making a new molecule, a new drug, is, I think, is very similar to the issue of Dr. Frankenstein and his trusty aide Igor making the perfect monster, right? So they've got to adjust the molecule, take a bit off here, add a little bit there, and to get it just right. So, you know, the chemists are splicing together new molecules that hopefully contain desirable features and eliminate the undesirable features. And the biologists then test them, and together they optimize and make the perfect medicine. Now, of course, in this team that I show you here, the chemists always see it this way around, with them being Dr. Frankenstein and the biologists being Igor, whereas strangely, the biologists always think that they're Dr. Frankenstein and the chemists are Igor, but the truth is, as with everything, somewhere in between. So let me just give you this is the only sort of chemistry, sort of scientific set of slides that I'm going to show you. But how did we get to this this drug candidate for malaria? Well, when we screened that original set of 5,000 compounds, one of the ones that came up as a hit, killed the parasite, was this one. And the final compound that's going to go into human beings in July is this one. You see, they're very similar. But down here, you have different parameters, a couple of parameters that talk about potency, a couple of parameters to do with solubility, a couple of parameters to do with drug metabolism. Green is good, red is bad, orange is somewhere in between. So you can see this compound didn't start off so clever, had a couple of green boxes. This one was amber, this one was red. These were so off the scale that we couldn't even give them a score. By making a very small change, changing that bromine to a fluorine, brought these other parameters on scale at least, so that was something. Changing this little group here for a different shaped group here gave us many, many more green parameters, and then finally changing this group here for this group here gave us the perfect or close to perfect molecule. So it sounds like 1, 2, 3 steps. But over 1,000 compounds were synthesized to get from here to here, because the degrees of freedom in chemical space are literally astronomical. So although it looks simple when you show it like that, the underlying work that has to be done by the synthetic organic chemist to make all of the myriad of compounds to get the perfect drug is very significant. Okay, so that's for malaria. Very briefly, what about tuberculosis? Because that's another disease that we work on. TB is an amazing drug, it's an amazing disease, a really ancient disease. And it comes as a shock to most people to hear that still 1.5 million people a year die of tuberculosis, a curable disease. So why is that? So over 9 million people each year become infected. And of those 9 million people, 500,000 get infected with what are called multi-drug resistant tuberculosis. So that's that resistance thing again. So this is TB that has become resistant to virtually all of the available drugs. So the first-in-line therapies for TB are really antiquated and very inadequate. They take at least 6 months to cure the patient, and that's the problem, because the patient starts taking them and actually feels better after about 6 weeks and stops taking them, and then the disease comes back again. And it comes back drug resistant because the bugs have been exposed to drug and then suboptimally exposed to drug, and that's how you get resistance. So we desperately need shorter, safer TB drug treatments. So again, I'm pleased to say there's a lot of international activity here. This is a horribly busy slide, but it just goes to show that a huge number of different agencies are working together to try and tackle this problem. So we play our small but significant role in all of this. There's the job of drug lead discovery, finding the hit. So there's a thing called the HIT TB Consortium, of which we're a significant player. That's funded by the Bill and Melinda Gates Foundation through the Foundation for the National Institutes of Health in the US. And then there's another organization called the TB Drug Accelerator Consortium, which, as you can see, contains a lot of partners, including several major pharmaceutical companies, as well as other academic centers. But about 20% of all of the synthesis of new TB drugs happens at the University of Dundee. It's not enough, really. It's a bit sad that 20% of all the world's activity on making new TB drugs occurs in a small university in Scotland when it's killing 1.5 million people a year. Nevertheless, this team is under Paul Wyatt's brilliant direction is making great advances, and the downstream partner for clinical development and getting it to patients is an organization called the TB Alliance, again, very heavily funded by the Bill and Melinda Gates Foundation. So that's what we're doing in TB. We're playing our part in a very large set of consortia. Lastly, what about bacterial infections? This affects all of us because a lot of these other diseases seem very far away in a developed place like Singapore. But what about bacterial infections? Well, drug-resistant infections, antimicrobial resistance, affects all of us. And this is from a report by Lord Jim O'Neill, who was the former Goldman Sachs chief economist, and he chaired a review on antimicrobial resistance. So this was really interesting. This is the UK government and the Wellcome Trust commissioned Jim O'Neill to review this area, and they chose an economist to do it rather than a doctor or a scientist. And I think that was a very smart move because he looked at it very dispassionately and very conservatively, and yet he came up with this conclusion that failure to tackle drug-resistant infections, bacterial infections, will lead to at least 10 million extra deaths per year and cost the global economy up to $100 trillion US dollars by 2050. 100 trillion. So maybe we should spend $1 trillion or $2 trillion on solving the problem before 2050 would be my suggestion. Okay, so when we think about antibiotics, we automatically think of penicillin because that's the quintessential antibiotic. So penicillin was and still is actually a wonder drug, but penicillin resistance emerged almost as soon as it was deployed. I mean, within months. Okay, that's the nature of antibiotic resistance. Now, this is my favorite poster. This is a poster which was put up in the factory, one of the factories in the US making penicillin. And it says, give this job everything you've got. Why? because penicillin, the new life-saving drug, saves soldiers' lives. Men who might have died will live if you give this job everything you've got. This is a World War II poster. But what I really like about it is here we have this recovering soldier sitting in bed enjoying a cigarette. Which just goes to show that not everything has changed for the better. Now, there's a little local story to tell about penicillin with respect to the University of Dundee as well, which is that during World War II, Robert Cook, who was our professor of biochemistry at that time, did this work. And actually this work was published in 28th of April 1945, even though the work was done in 1942 and 1943. This was a trade secret, a wartime secret. It was D-notice was put on it by the UK government because this was such a discovery of such importance to the war effort. And what Professor Cook discovered was that green pea juice is a fantastic medium for growing the penicillin fungus to produce penicillin. And as I say, it was published in Nature. Some of you here will know the value of publishing in that journal in April 1945. So back to penicillin. I said it is and kind of was a wonder drug. Why it was? Because penicillin resistance is really rife. So there's many, many bacteria infections which don't respond to penicillin anymore. So why do bacteria become resistant to drugs? Well, if you have a population of bacteria and you attack them with antibiotics, you may have one or two resistant bacteria in that population. So you kill all the sensitive ones, but the resistant ones are left behind. And as that population expands, now you have a big cloud of drug-resistant bacteria. And these are the guys that are going to infect the next poor soul. So it's a really simple problem that we have with bacteria. There are many different ways that bacteria can be resistant to drugs. Some bacteria simply don't allow the drugs to get in the first place. A lot of so-called gram-negative bacteria fall into this category. Some bacteria secrete proteins which either bind the drug and inactivate it or break the drug down into harmless little pieces. Some bacteria, the drugs get inside the bug, but the bug pumps them straight back out again. This is a major problem, efflux mechanisms. So there are lots of different ways that bacteria can become resistant to antibiotics. So the trick is to keep on making new ones. If you keep on making new ones, you can stay ahead of the bugs. So when they become resistant to antibiotic A, penicillin, you can treat the infection with erythromycin. So that should be no problem, right? Well, here's the problem. This was the golden age of antibiotic discovery between the 1940s and the 1960s, mid-1960s. All of these different classes of antibiotic were all discovered and developed in that time frame. A couple of new classes came online in the early 2000s, but there's a massive innovation gap where we just stopped making new antibiotics. Why did we do that? Why did we stop? Well, it looked like we had plenty. So, and we could keep on tweaking these a little bit, put a methyl group here and a hydroxyl group there, and keep on, refreshing them. And it was thought, well, we've got a great pharmacopoeia of antibiotics, should be no problem. But it's that dead zone of antibiotic discovery that's left us in this parlous state now, because the bugs have become resistant to these whole classes of antibiotic through overuse. People like ourselves going to the doctor with a sore throat and say, oh, doc, I've got to have an antibiotic, even though it's a viral infection. All right? From agriculture, in parts of Southeast Asia, they use animal powder. What's that? It's a mixture of 20 antibiotics that are in human use that they sprinkle on their animals to make sure they stay healthy. So the amount of antibiotics in the environment is colossal. And if you have antibiotics and bacteria in the same place, you're going to get antibiotic-resistant bacteria. Then they get into the sewage system. They can transfer their information on drug resistance from one bacterium to another, so it spreads throughout whole populations of bacteria, and then guess what? We get infected with them, and then we go to the doctor and find that none of the antibiotics work anymore. Very serious problem. So what do we need? Well, obviously we need new antibiotics, right? If all the current ones are failing, that should be a given. But we also need antibiotic stewardship, the better use of existing and future antibiotics to prevent the spread of resistance. And of course, like everything else, we need better diagnostics. So what are we doing in that regard? Well, my friend and colleague, Professor Dilip Nathwani, who's a consultant in infectious diseases at the University of Dundee, is actually very, very active in this area. He's a big expert in antimicrobial stewardship. He works for the National Health Service of Tayside, part of the Academic Health Sciences partnership with the University of Dundee. He's also currently president of the British Society for Antimicrobial Chemotherapy. And as I always say, when it comes to antimicrobial stewardship, he's the big cheese. And Dilip's favorite quotation is this one from Dennis Mackey, which is, "The development of a new antibiotic without having mechanisms to ensure their appropriate use is much like supplying your alcoholic patients with a finer brandy." And he's absolutely right. So people like myself and my colleagues who are at the sort of basic science discovery end, we definitely need to be making new classes of antibiotics. That's our job. But at the other end of this spectrum, at the clinical and agricultural end, better surveillance, better stewardship of the use of antibiotics is absolutely vital. And here's an example of what you can achieve when you do get it right. So this is one of Dilip's colleagues, Dr. Karis Marwick, and she's been involved in stewardship interventions. And you can see here they introduced stewardship interventions back in 2007, and by 2014 the amount of antibiotic-resistant Clostridium infections had dropped remarkably. So this led to less antibiotic resistance and better clinical outcomes for the patients. Not by using more antibiotics, but by using less antibiotics. That's the whole point. We can get better clinical outcomes with less drugs, not with more drugs. So when you go to the doctor next time and she or he says, "I don't think you need any antibiotics," please do take her or him seriously. And one of the great things that Dilip and his colleagues have done is they've created a MOOC, a massive open line course on antimicrobial stewardship, which has got 220,000 people registered, over 8,000 active learners. I've done the course myself. It's absolutely fascinating. It tells you, it's really designed for clinician experts. It tells doctors what they need to do to spare the use of antibiotics, to make the antibiotics more useful in their clinical environments. So it's a great effort by Dilip and his colleagues. And in the last minute, I'll just tell you what else we're doing in Dundee. We've created a thing called a Centre for Antimicrobial Resistance, and we're bringing together the expertise of the Drug Discovery Unit that I've told you about, but also our clinician colleagues, our physicists and mathematicians, and our molecular microbiologists to try and work together on this problem. And one of the things as a component of that is we're creating an antibacterial drug discovery accelerator. We can't deal with surveillance or diagnostics or stewardship in life sciences, but we can deal with the treatments part. And the idea here is to take discovery science from all the great microbiologists there are not only in Dundee but across the world, and help them in this void bit to get their targets that they discovered into lead drugs. Now, just at the moment, there's a lot of activity in taking drugs which are stalled in the pipeline and getting them through to clinical, preclinical and clinical trials, and that's great. But once that's done, there's a real void in the pipeline, and we want to try and help our basic science colleagues to get their targets moved through to a position where they can be moved forward into clinical usage. So that's one of our current goals. As with everything, it needs funding, and we're fighting like crazy to find the funds to get that activity off the ground. And there are lots of different innovative pieces of discovery science that you can use to tackle this problem. One is biofilms, for example. So what are biofilms? Biofilms are those slimy things that you slip on, right? You know, when you're walking along and you come to a slippery patch, that's usually a community of microbes in a biofilm. It's bacteria mostly joined together by a sticky glue that they exude to help them become very resilient. And for example, my colleague, Professor Nicholas Stanley Wald, brilliant young scientist, has brought together a consortium of other scientists from mathematics, from physics, from imaging, from engineering, even drawn in one or two bog-standard biochemists to help try and sort out the fundamentals of biofilm formation. Because actually there are lots of clinical infection situations where the bacteria themselves are sensitive to antibiotics, but the antibiotics can't get to the bugs because of the biofilm. So if we understand how biofilms are formed, that may be a new and innovative way to tackle infection. Just one of many possibilities, just putting one up there for an example. Now, I think I've rattled on for far too long, so I'll just stop there. But I'd like to make some acknowledgements. And first of all, I'd like to thank all of my colleagues across all of the sectors of the University of Dundee whose work I've drawn from for this presentation. Obviously, I'd like to thank all of the funding agencies who supply us with funding to help tackle these diseases. And that's because it came from a different talk, so I'll skip that. And I'd just like to thank you all very much for being here and listening to me. Thank you very much.
Okay, we're now going to open up for questions. If you could, please keep the questions short so that we can get to as many people as possible. So if you put your hands up, we'll try and get a mic to you, and if you can tell us who you are and where you're from. So we'll start now. Sorry. Looking at that.
Thank you. You said that the development of this mucil transferase inhibitor for Kinetoplastid treatment was abundant because it didn't get through the brain-blood barrier. Yeah. Let's say lumbar injection maybe is not a very convenient way of treating, but in the case of a deadly disease, maybe better than dying.
Yeah. No. Actually, we also did re-engineer the compounds to become penetrant. But then we picked up neurotoxicity, and that's what really killed it.
Okay.
So it's a fantastically safe drug for treating the systemic infection, but it wasn't a good mechanism for treating the neurological complication. Yeah. Sadly.
Thanks, Prof. Ferguson, for the talk. I want to follow up on the previous question as well. I was wondering, because you mentioned that this could not affect stage 2 trypanosomiasis, right? I was wondering whether these N-myristoyl transferase inhibitors could be used in prophylaxis.
Yeah, they could actually. And one of the things I keep on reminding my friends at DNDi is that actually it'd be, the aim in future is to never roll out a monotherapy because you get resistance very quickly, but to have a minimum of dual therapy. It's very hard for an organism to become simultaneously resistant to two things. And actually, the N-myristoyl transferase inhibitors would be pretty good, very efficacious protecting component to a dual therapy with something which is blood-brain penetrant, because the tsetse flies don't feed from your brain. So actually, I think if these compounds have any clinical utility, it will be to roll them out as a dual therapy to protect a stage 1, stage 2 penetrant drug.
I see there's a lot of research to develop new drugs, but why is there no research to kill the bacteria at source, eliminate them totally? So that's one question. Second question is, decades ago I heard about the TB was already eliminated, but subsequently another news came out saying the TB came back in a big way.
Yeah.
What happened?
Okay,
Thank
so
you.
TB was never eliminated, I'm afraid to say. It's always been very prevalent on the planet, and just as prevalent now as it's ever been, actually. So that one never has. So eliminating a disease-causing organism is difficult. It's not impossible, right? So smallpox has been eliminated, and polio will be the next. But you need to have good surveillance, good diagnostics, very safe and efficacious vaccines or drugs to do it. So, one of the goals at the moment is to eliminate malaria. There's two words, eliminate and eradicate. They're different. Eradicate means gone from the planet. That's difficult to do. Eliminate means bring the number of human cases down to zero or virtually zero in the region that you're talking about. It should be possible, we hope, fingers crossed, to eliminate malaria in maybe the next 20 years by having the right combination of prevention, which is bed nets, and the right drugs, which are efficacious. And one of the things that Medicines for Malaria Venture is aiming to do is to create a thing which is referred to as a single encounter radical cure and prophylaxis, CERCAP. What does that mean? It means a child will turn up at the clinic with a fever. You'll first test with one of those finger prick type things whether she or he's infected, and then you'll give the child one pill and you'll watch the child take it, and you'll know that child will be cured, and that child will be protected against further infection for at least 2 weeks, and that child will be unable to transmit the disease to any mosquitoes that happens to bite them. That's what we're aiming to do. I think it's entirely doable, and when we have that tool, plus the bednet protection systems, it should be possible with the right public health interventions and the right delivery systems to eliminate malaria from large parts of the planet. That's the goal. So it is doable, but it's difficult and it requires coordination of many things.
There's an opinion that the development of new antibiotics is very much slower now because the drug companies are not interested in it as much compared to a cancer drug development, because cancer drug is a very big market. Yeah. Antibiotics, you can make money. Yeah. Do you agree to this?
Yeah, it is absolutely true. So one of the reasons why many pharma companies have pulled out of antibiotic research over the years, AstraZeneca, for example, pulled out completely 2 years ago, is because the business model doesn't work for them. So what they say, and they're absolutely correct, is if we develop a new broad-spectrum antibiotic, you, the clinical world, are not going to use it. You're going to leave it on the shelf as a reserve drug, and therefore the number of copies of this drug that I've just spent $2 billion making is going to be is very low. So the turnover is too low to make any recompense for the investment. And they're absolutely correct about that. That's exactly why they've pulled out. So, but it seems to me that as a species, we do need these things. And therefore, our politicians and our governments, G20 for example, need to be figuring it out. How do we make a new recompense system so that people are incentivized to make new antibiotics? And that's what the Lord O'Neill's report is all about. It discusses various different financial models whereby it may become worth the while of the pharmaceutical industry to get back into antibiotic discovery. In the case of malaria, you know, it's a great example. 15 years ago, most companies 15 to 20 years ago, most companies pulled out of any anti-malarial development, saying it was too hard, too difficult, not enough profits in it. And yet this PDP, Medicines for Malaria venture, on a very slim budget of about $70 million a year, over 15 years, have come up with a whole pipeline of new drugs which are going to solve the problem. So the amounts of money involved don't need to be that massive, but there does need to be a correct economic system to make it worthwhile for commercial partners to play their part. And I'd like to think that this isn't beyond the wit of man to get it sorted out, but it will require a lot of international cooperation at a time where international cooperation isn't at its highest. But you know, it has to be doable. We can't afford not to do it. It has to be done. So meanwhile, while the great and the good sort all of that out, people like ourselves, basic scientists, will just get on with the job of trying to find some new classes of molecule that might be useful in the future. So, and there's a big activity worldwide in this area now, and a growing interest amongst basic microbiology experts to take their discoveries and let them be translated towards new therapeutics. So whilst it's very, very serious, again, I'm not pessimistic. I think it's an entirely doable and solvable problem. We just need to get on and get started and do it.
I hope this isn't too controversial, but we've heard a lot about fake news and so on. And we hear more and more about, I don't know, well, I mean, you see how ignorance has to be fought through stewardship and so on. But, you know, the MMR vaccine, as we know, has been a growing concern about this, I mean, whether it's real or not. So do you think, is that going to be a bigger problem, or is it a big problem, or are you worried about it?
I think if you're in hospital with septicemia, you're going to want to take an antibiotic. So first of all, with infectious diseases and very efficacious chemotherapeutics, I just don't think there's much of a debate, because, you know, people are very, very seriously ill. They take the medicine, and 48 hours, they're better. It's pretty convincing. So I think that really is important. With vaccines, you talk about the MMR case. This is really dangerous because at the moment there's a huge backlash against vaccines. There's this issue about, does MMR vaccine cause autism? I can tell you that it does not cause autism, right? There's been a huge amount of excellent science done to show that that is not the case. And yet this myth persists in the media, stoked by disreputable scientists and doctors who've been struck off and are really doing the world a great disservice. Because of that kind of publicity, there's a huge number of people now, understandably, who are very scared about having their children immunized. There's a measles epidemic in central North America at the moment because of this. Parents scared to death of having their children immunized because of the MMR vaccine myth. Haven't had their children immunized, and now there's an outbreak of measles. Well, some of these kids are going to be deaf and brain damaged. We can't afford to let ignorance trump scientific evidence. So I'm really, really sorry about the pun. I'm really adamant about this, and it's beholden on all of us, all educated people, to make sure we get the right evidence to the population and explain to them, what the importance is of things like vaccines.
You were talking about microbiologists inventing new drugs. Hypothetically, if I'm a microbiologist and I have this new drug candidate that I've been developing in my lab, what is my pathway from here to there?
Yeah, no, it's a great question. If you're a microbiologist working in the lab, you're not so much likely to have a candidate drug, but what you probably will discover is a drug target, some physiological fundamental process of the organism, which you discover, if we could only just stop that from working, we'd kill the bug. From there, you need to then partner with an operation like ours, for example, a drug discovery team that will say, okay, from that we can develop an assay which we can screen hundreds of thousands of compounds against, and when we'll take those hits and work with you to find the best ones, and then do the tweaking, the medicinal chemistry tweaking, to make them better and better and better. But that's really what it's about. So the discovery scientist has a huge role to play here, because it's the discovery scientists who will find the new targets and pathways that the drug discoverers can then work on. It's a true partnership. And in the way we operate, we always work very, very closely with the initiating biologists all the way through the process, because we don't want to lose their expertise going from the beginning to the end. You're welcome.
You mentioned that there is a difference between elimination and eradication of diseases.
Yes.
So are there any diseases which have been fully eradicated? If not, okay, wait, could you give some examples of them?
Well, so the disease that's thought to be eradicated is smallpox, but a vial of smallpox virus turned up at a British university once after eradication had been announced. So it's very difficult to know when you've eradicated something, because the proof of non-existence is very, very difficult, okay? So I would say, I don't like to use the word eradicate diseases, because I don't think it's a very practical statement. But to eliminate diseases, to bring the number of human cases down to 0 in any one given year, is entirely doable. I don't think there were any cases of smallpox last year, for example, or the year before that. So it's definitely eliminated. But if you're deciding which word to use in discussions about disease, I would just advise you to use eliminate, because eradicate is really a tough thing to do.
Okay, thank you.
You're welcome.
I'm just curious about one thing. We've always been discussing about antibiotics and how it kind of like kills bacteria and stuff. But can we like develop the human body to like make their own immune system to be able to cope with the evolutionary?
Can you put your hand up, because I can't see you? There you are. Thank you. Yeah. So can we treat with drugs which alter how the body interacts with the microbe? You mean?
I'm curious, because like we as a human species actually constantly evolve and grow. So why can't we like make substances that can actually like alter our immune system behaviors to the point where we can actually adapt to kill the bacteria? Is it a possible means to solve the problems?
Yeah, and so in fact, actually, vaccines do exactly that, right? So we take a vaccine, and then we become completely immune to the microorganisms. So all we're doing is we're teaching our immune system to recognize the bug before the bug gets to us. So in many respects, what you refer to is vaccines. But in a similar vein, there's also a lot of discussion about drugs which modulate our immune system to battle infection. So, you know, if you get infected with a microbe, it's because your immune system has failed to deal with the microbe. So maybe there are some things you can do to tweak our immune system to make it, you know, super active against the bug. And so there are many people working on those kind of solutions. The only thing about the immune system is it's a double-edged sword. Often if you switch it on too well, you get autoimmune damage to the to yourself. So you have to be careful, but it's certainly not impossible. But vaccination is one such example. Thank you for your question.
What's the impact of neuroscience that you see? I mean, definitely, I mean, the failure of your previous drug, that's a great example. But in terms of like drug discovery, it's probably a huge challenge, because instead of looking at cells that are normal cells, neural cells are kind of quite much more sophisticated. Yeah.
Yeah, no, I mean, making drugs for neurological conditions is very difficult, and multiple problems. One is the molecules have to be of a particular design to get into the brain at all to begin with, and then the brain is a very sensitive machine, and it's very easy to get neurotoxicity and make people terribly unwell. And for this reason, actually, the majority of pharmaceutical companies have also pulled out of neurological diseases over the years, right? Because they've spent a lot of money trying to do it and failed. Failed for all the right reasons, not for the wrong reasons. And part of the problem is lack of models for human neurological disease. So sometimes they can make drugs which cure the animal model, and then they find it has no efficacy whatsoever in human beings. This is very difficult, because unless, you know, we can't give experimental neurological drugs to human beings, right? Can't do it. It's not ethical. So it's just a huge problem. So I'd like to say something soothing, but I've really got nothing soothing to say about it. Yes, and who knows what kind of technologies are coming along. I mean, here at A-Star, there's fantastic work done on stem cells, for example. And, you know, there's a lot of development of you know, liver on a chip, lung on a chip type technology, creating micro versions of organs. Who knows? Maybe we'll have a brain on a chip one day, and we could do a lot more research on neurological defects before we test them on humans, but using human neurological tissue rather than animal neurological tissue, which is what we have to do with animal models. So yeah, it's we're a long ways off having all the right tools for neurological diseases, sadly.
Hi, I've got a question about one of the Kinetoplastid diseases that you mentioned earlier. Sorry, I'm over here.
Oh,
following on from the question about engineering the human body, so you said that one of the Kinetoplastid diseases infects macrophages and attacks them from the inside. So would there be a way to combine kind of genomics, research into genomics, to kind of edit the macrophages in some way, the proteins that they express, to stop them getting inside the macrophages? Or are there too many kind of unknowns involved in that for something like that to happen?
It's a really, really interesting question, and in fact, I would say that that is technically possible. The problem is how we're going to edit the genome of everybody's macrophage, if you see what I mean. So, but technically, you ask a really important question, because what you're asking is, what is the role of gene editing in human disease? Actually, is the question that you've asked, and it's a really profound one. And if you perform a gene editing of the germline, then of course not only that, the progeny and all that person's progeny could have that same genetic makeup. So it's technically possible that you could make a line of human beings that were all resistant to leishmaniasis. But to roll that out across the whole species would be kind of difficult. But here we have the dilemma of germline gene editing becoming a plaything of the rich and not available to all. There's a huge set of ethical issues to be discussed there, and I'm very glad that you brought up that subject, because that's maybe something we can all discuss over some nibbles and drinks later on. It's a really important point that you brought up. Thank you.
Okay, I think we can take one more question.
For TB, right? Every Singaporean is given a BCG vaccine.
Yeah.
So since we have antibodies against TB, I don't see why is it so prevalent in Singapore.
Yeah. Sadly, the BCG vaccine doesn't work for everybody. So there's very good evidence that actually it works pretty well in Caucasian Europeans, but there's very little evidence that it works in much of the rest of the planet. And I don't know what to say about it other than we don't have a vaccine that works. And there are experimental vaccines being produced at the moment. There was a clinical trial just run on a very sophisticated one in South Africa 2 years ago, and that clinical trial just failed as well. I mean, it's a really high-tech vaccine, had all the right hallmarks. It should have worked, but it didn't. So we're still in a position where we don't have a highly efficacious vaccine that works against tuberculosis, sadly.
Okay, ladies and gentlemen, please join me in showing our appreciation to Professor Michael Ferguson. Now, for all the students here, if you are interested in life sciences at the University of Dundee, there are some admission staff here. So if you want to stay behind and ask them questions, they're more than happy to help you. But for everyone else, there is a networking reception outside. So join us for that. Thank you.
Hey, shit, everyone else is there.